Welcome to the PharmaSource podcast.
The journey from academic research to clinical application requires navigating a complex landscape of GMP manufacturing, regulatory requirements, and quality standards. A transition that presents a significant challenge even for the most innovative research teams. Today, we're joined by Dr. Jenny Prange, Head of Project Management at Wyss Zurich, Regenerative Medicine Technologies platform, RMTP. So Wyss Zurich is a unique accelerator embedded within ETH Zurich and the University of Zurich, consisting of two platforms, biomedical and robotics technologies on the one hand, and the Regenerative Medicine Technologies platform on the other. Wyss provides office space, GMP manufacturing facilities, and expertise specifically designed for university spin-outs and early-stage biotechs working in regenerative medicine, robotics, and advanced therapy medicinal products. Jenny is responsible for guiding projects at the RMTP through their critical first steps into clinical manufacturing, from setting up proper analytical methods and process validations to securing regulatory approvals for clinical trials with Swiss authorities. In this episode, we'll explore the specific manufacturing challenges that emerge when translating academic research into clinical-grade production, why materials and processes that work perfectly in R&D labs often can't be used for human applications, and how early-stage biotechs can build robust CMC strategies that satisfy regulators and attract investor confidence. Jenny, thank you for joining me.
Thank you for having me.
So could you just start by telling us a bit about your background and also what Wyss Zurich actually does?
Yeah, certainly. I did my bachelor's in molecular biotechnology at the University of Dresden. And afterwards, I moved to Strasbourg for my diploma thesis in biotechnology again at École Supérieure de Biotechnologie de Strasbourg. And then did my master thesis at the Novartis Institutes for Biomedical Research in Boston before I eventually moved to Zurich in Switzerland for my PhD in human physiology. And shortly after that, I started as a head of production in one of the projects that we have at the Wyss Zurich. And my task was to translate that specific R&D process into a GMP-ready production for clinical trial. And in the end, also to conduct the following phase 1 and phase 2 trials at the University Hospital in Zurich. So this is how I got in touch with the Wyss Zurich initially in 2017, so it's a while back already. And it was mainly as a platform where we could translate our research process into something more regulated and ready for clinical production, which is exactly what I'm also focusing now in my new role as project manager, where I can leverage the experience that I gathered through the project time that I had before. And while the Wyss Zurich celebrated last year its 10th anniversary. And over the years, Wyss already supported 28 projects by what you said already, offering funding, not only for translational R&D, but also for early stage clinical phases, like we did before, and coaching and mentoring by subject matter experts, and also giving access to the different projects to technology platforms and the infrastructure. So like this, the regenerative medicine platform has clean room capacities where those projects can perform the manufacturing for those clinical products that they want to develop.
As a really interesting sort of niche within the space. So you primarily work with university spin-outs and early stage biotechs. What's the biggest knowledge gap you see when these projects first come to you?
What comes to mind primarily is the regulatory requirements, because those build the basis for all the workflows that you want to translate. And you don't only have that on the clinical part where it's obvious, right? Because clinics is working with humans, but also on the manufacturing side. So projects quite often realize only when they started with us that their R&D process cannot just be copy-pasted, do it in the lab, into the manufacturing facility. And that there are rules that they have to follow to bring something into a human trial. And since most of the members of those projects are purely of an academic background, they have never heard from the IMPD or of the IMPD before, or the IB or the study protocol before they come to us, right? So that understanding that those super important, but also not short documents are the basis for setting everything up is just not there.
Really interesting. So what does surprise the most about that sort of leap you were talking about then from academic lab to GMP manufacturing? Obviously, many of these researchers have excellent preclinical data, but yeah, what surprises the most about that next step?
So what comes from that regulatory gap is that there's loads of documentation that you have to submit to authorities when you register for a clinical trial. And to have the documentation ready, that costs a lot of time and resources. And this is what is always underestimated. Also, the sheer time that it takes to translate a process from academia to clinical manufacturing is just highly underestimated. There's a lot of people that have no idea whatsoever how long it actually takes to translate a process. And it was the same for one of the projects at Wyss Zurich, where the initial process was supposed to be translated within half a year. But in the end, it actually took 2.5 because there were several gaps that came one after the other, and you had to fulfill them before you can go into clinics.
That really is a long timeline. If you thought it was going to be 6 months and it ended up being 2.5 years, where was the sticking point there? If it's a long process, why would you expect it to be 6 months? Like, where's the gap?
The gap is that when you set up the process, you have to prepare loads and loads of guiding documents or also work instructions that or the operators that go into the clean room have to follow in the end. And there's just the understanding and the knowledge of how long it takes to generate those documents and then also test them. It's just not there.
What steps need to happen to prevent those delays? What do you need to have in place before to reduce the risk of delaying a project? Or is that just part of it? A delay is just part of the process and there's nothing you can do?
No, you can do a certain amount, I would say, but there will be always a delay because it just takes time to translate a process, because the basis of it all is also how well was everything documented in the preclinics. So, and I know from experience from my academic background that the interest of supervisors, how well you document your experiments and your results, ranges from not interested at all to, well, slightly interested, as long as you can generate a graph that is presentable and the publication in the end. So if that would be somehow streamlined, that would probably help, because it emphasizes already the importance of the basis data that now I see also more and more in peer-reviewed papers they request when you submit publications. And this is exactly the data that you will have to submit in the end also to the authorities to get something approved. And the better that is documented from the start, the less delays you have in the end.
So how hands-on is Wyss Zurich in helping projects navigate these regulatory approvals?
Wyss Zurich organizes meeting opportunities, such as the innovation office meetings with Swissmedic, so the Swiss authorities, on a regular basis, where current and also future projects can present and discuss their processes, their strategies. They can ask the regulators specific questions, for example, how they want to approach their CMC strategy, or what kind of additional study they would need to perform before they're accepted for clinical study. And they get a reality check, basically, directly from the authorities. So that helps a lot. And then also Wyss Zurich obviously helps in getting support in when, for example, documents like the IMPD, which is the basis for the manufacturing process, right, that has to be translated, comes, and it becomes obvious that it has been either worked on from 20 different people because it's repeating very important sections and then having copy-paste errors in the whole document, or when there is no experience whatsoever in how to write the documents from the research side, because those documents are not like a scientific protocol that you can just follow. They are well structured, but it's a certain way of writing, I would say, that people have to get used to. And this is where the Wyss Zurich obviously helps also the projects. But in the end, the responsibility of submitting all those documents and having the interaction with the authorities, that lies with the projects. But there's also other institutes like the Clinical Trial Center, for example, here in Zurich, that can support in those cases.
Interesting. Let's move on to talk about manufacturing sort of as a de-risking strategy maybe for projects. So how does having clinical trial manufacturing capabilities make a biotech more attractive to investment?
When startups or projects come to the regenerative medicine technology platform, they're very early stage, right? So that means they have a couple of proof of concept studies and tox studies performed, and a very well thought through strategy how this drug eventually can hit the market. But if they would approach investors with just those ideas, they see a huge risk associated with that because you don't have preliminary data how that would work in humans, for example. So the Wyss Zurich comes into play because they can offer the projects the chance of gathering that so important first clinically relevant data. And with that, they can reduce the associated risk for the investors. And quite often, projects tend, or like first phase clinical trials, which are primarily focused on safety and not efficiency of the product yet, projects or PIs try to include already as maybe secondary endpoints, preliminary efficiency readouts, because they know investors would like to see them, that they can present something, although the first phases are actually not foreseen to support such a claim.
At what stage should biotech founders start thinking seriously about their manufacturing strategy?
Well, they should start thinking about it as early as possible, obviously.
Yeah.
But from our experience with different projects, I would say the first phase can always be performed in a very close state of how the process was developed in academia, so in basic research. But as soon as you have the first clinically relevant data, and it's been shown that it's safe, that should be the point where the improvement of the process should also be tackled, because eventually you don't want to have too high cost of goods when you manufacture a product, right? And then you can critically assess also if there's other options. For example, if you started your process with full setup, if it's possible to somehow automate this or introduce certain machines that can either reduce the time that the process takes or also the risk of operating steps that have to be performed manually.
So walk us through what typically happens when a university spin-out comes to you with a promising therapy. Where would you start?
First of all, we have a look at what the whole idea is and how the process looks like. And then we check how this, because it will be an R&D process that they present to us, how we can integrate this in our facility and what the scope of the whole process is and what are the critical points that we foresee already and how we can control the impact of those critical points, that this will not be the reason why in the end the process will be failing. And when we have a closer look, then we start obviously with the materials, so that were used in the research lab, and which of those materials potentially have to be replaced, or with a high probability, or which of those materials miss stability data. For example, that will happen quite often with the starting material. If, for example, processes start with like a blood product or a tissue sample, for example, then quite often it has not been defined how long those materials are stable, or so basically what the shelf life is. It's not the same if you buy a buffer from a known provider, they give you an expiry date for that. But if you get, like, for example, fresh tissue, you have to generate the data yourself. And most often people are not aware that this data is actually needed to define an expiry date. So we decide what of the materials that we need for the process are critical, maybe which of those need additional testing to be declared safe and the appropriate quality for a clinical trial in humans. Then obviously the process itself. So how well is it characterized? How detailed has it been documented in the research, or what important inputs are missing, for example? And quite often there we also see a huge difference in the protocol from the R&D, which tends to use to be 1.5 pages or 2, like for the whole process in total. And then once you translated this, it ends up with 8 separate documentation instructions, right? And then also what is very important is the different assays you have to include in the process, because you have to characterize your product somehow. Then the question is which of those assays will be in-process controls, which of them would be release assays. Quite important there is also how fast can those assays be done? So how fast do you get the results in terms of how fast can your product be released in the end? And also with those assays, an important point would be which of those assays can be regarded as a potency assay in the end, because authorities tend to have a look at that also quite early on. So for those meetings that we set up with the Swissmedic is also helpful, because like this you can interact with them from an early stage onward, because with the ATMPs, every product is a bit different. So you have to come up with your own potency strategy. And if you get the input from key opinion leaders or the authorities, that is very helpful in those stages. Well, and obviously, once you decided on the assays that you want to run, you have to verify them to make sure that they always work out and that you can rely on the results that you get. And in the end, you piece everything together and do a couple of technical batches to make sure everything works. And once this is smoothed out, then you run your process verification, which basically fixes the process for the clinical stage. And then as a last step before the clinical preparations can start, you have to do an aseptic processing, so media film, what is called, to validate the whole process and the operational personnel that is performing the process in the end.
Let's go back to where you were talking about the materials and reagents sort of used in R&D labs versus what's needed for clinical manufacturing, because it can be quite different. What are the key differences, though, that biotechs need to understand?
The classical situation is that material that is used in the R&D labs is usually also R&D grade. So those materials, when you ask manufacturers, most likely don't have any certificates where they prove they have been tested for certain components or analysis types. And this is the stuff that you need when you do your clinical productions, right? Because those certificates are the basis to prove that the quality of the material that you want to use in your process is adequate for clinical production. And when you have materials that have those certificates, you reduce the risk, because then you can maintain a certain quality standard for the whole process. And our task is mainly that we make the projects understand that any material or component that is entering the GMP process needs to be as clearly defined and controlled as possible in order to reduce any potential risk that could come from that. So there we need certain acceptance criteria. And for this, we need, for example, buffers or buffer components. We have only a sterility certificate, but for example, no information on endotoxins or mycoplasma, for example. Then we have to think about how we can introduce an additional testing of the medium before we can use it in the clinical production process. So there we sit together with the projects and assess what are the materials, what we need to replace, and what we need maybe in a higher quality grade, because sometimes manufacturers also offer the same type of material but in different grades, so that the research material is not as expensive as the GMP grade because it has been tested.
That was kind of going to be my question. Why can't you just use manufacturing grade materials in R&D? Is it just the cost factor?
It is mainly the cost factor, yes. Research is running on public funding quite often, right?
Yeah.
And then you have to probably count and consider very carefully which antibody you order. And there's a huge difference if you order a medium, for example, that is RD grade or a medium that is GMP grade, because quite often that's not only a duplication of the price, it's much more.
I see. Yeah, makes sense. Right. So how do you see the role of academic or translational manufacturing facilities like yours, like Wyss Zurich, fitting into the broader CDMO landscape?
So I think the role that we can take is we can try to fill the gap that there is because of for example, the regulatory knowledge that is not there, because most of the academic teams have never really considered what is needed. And I can also say from my experience, from my studies, that we all heard the different stages for clinical trials, but we never went into depth of each of them or what it would mean to really start one. So there's just no idea behind that. And I think what Wyss Zurich can do is we can help translate those preclinical research ideas into a first GMP-ready process, and also have the project to perform the first phase clinical trials. And then once all of this is done, we basically had the projects already in that very important first tech transfer, which in my opinion takes also most of the time, because that is just a higher hurdle to jump from R&D to first phase clinical trials than what you have to jump from a first to a second phase clinical trial. So, and we just basically set the groundwork for that, and the projects are done. we can also give them basically a pack of documents to go to the next CDMO, for example, if they have the required funding for that, and say, hey guys, this is our process. We need to set it up here. And this is how we did it before. And that just goes much smoother than as if you would come with a R&D process and say, this is what we thought of. Can you help translating this?
Right, so you're sort of a halfway house for projects. Once a project successfully completes a trial with you, then what would typically happen in their manufacturing journey? They move to commercial CDMOs or build their in-house facilities.
There's actually both things that can happen, and that happened already. So what it is, in the first place, they phase out. So as a kind of academic manufacturing facility that we are, as I said, we gather the documents and prepare it, and then either hand it over to the project themselves if they don't have decided yet where they will go for the next manufacturing step, or we hand it over to another CDMO. And there was also a case of one of the former projects. They built their own GMP manufacturing facility, and they did it still while they were at Wyss Zurich. So in that time frame, they even did then comparability studies to show that the process works equally well with us and then also in their new manufacturing facility.
As a closing note, if you could give one piece of advice to a biotech CEO who is just starting to think about their CMC strategy, what would it be?
So I would say that should be considered as early as possible. And I think they should also consider sparing the necessary resources available. So meaning that they hire a person to focus on exactly this. Yeah. And because those questions are just so important that they should not be tackled on a side note by, for example, people that focus on manufacturing only. And then on the side, they are still working in an R&D lab, because they need to critically assess the process and the characterization assays. And they also need to gather external advice, not only from key opinion leaders, but also in very close exchange with the authorities to get feedback, what is expected, and also, on the other hand, to check what is realistic for their specific product. Because, as I said before, the ATMPs are just so different. There most likely is no one solution fits all kind of approach. You have to come up with your own. And for this, you have to understand what are the requirements, and you have to also know what is possible for your own product. So I guess it comes down to just build the relationships with authorities as early as possible.
Cool. Well, thanks a lot, Jenny. I really appreciate you taking the time out of your day to chat to me about this.
Thank you.